Hi, I'm Dr. Piero Perucca, and today I will be talking about the potential of drug repurposing in epilepsy treatment. It's important to start with definition, and I'll provide a definition of drug repurposing or drug repositioning using the NIH definition. So repurposing generally refers to studying drugs that are already approved to treat one disease or condition to see if they're safe and effective for treating other diseases. The process of drug development is a long and expensive process. It may take up to 16 years and can be as expensive as $2 billion. Drug repurposing or repositioning offers a shorter time scale, so 6 years, and is much less expensive, on average $300 million. Examples of drug repurposing are available every day in that anti-epileptic drugs are often used to treat other conditions. Today, we will focus on drugs which are approved to treat other conditions to see if they are effective in the treatment of epilepsy. Over the years, many anti-epileptic drugs have been introduced, and the latest compound being fenfluramine, which was approved just last month. We have now 31 drugs, many of which were introduced after the mid-90s, resulting really in an explosion of novel agent and in an expansion of the arsenal available to prescribers to treat their patients. The question is, have these drugs made a difference in the outcomes of epilepsy therapy? And to answer this question is this cohort, which has been very well studied, the Glasgow cohort, led by Martin Brody, can really provide an insight. Everyone is familiar with the landmark study by Patrick Quine and Martin Brody in 2000, which included 470 patients with newly diagnosed epilepsy who were followed over time following commencement of anti-epileptic drug therapy and changes according to treatment response. Well, 64% of these patients became seizure-free over time, as opposed to 36% of patients who had uncontrolled seizures. So the same authors reappraised their expanded cohort to see if the introduction of new anti-epileptic drugs actually have made a difference in the treatment of epilepsy. And in a cohort that now almost comprised 1,800 patients, the outcomes were remarkably similar to the original description. So 64% of patients were seizure-free, and again, 36% of patients continue to have uncontrolled seizures. So the treatment of epilepsy has not benefited from the introduction of new agents, and this is fairly disappointing. What we've witnessed, however, have been major advances in understanding the mechanisms of epilepsy. This include a better understanding of molecular pathways, of cellular mechanisms, and an important role in this space has been neuroinflammation, which has really emerged as having a critical role in the process of epileptogenesis. Neuroinflammation has definitely a role in a form of epilepsy which has emerged and has been better defined over the past few years, which is autoimmune epilepsy, which provide also a great forum for to display the efficacy of drug repurposing, which is in the form of immunotherapies. This is a well-described study in cohort by Toledano et al., published in 2014 in Neurology, who studied 29 patients with autoimmune epilepsy. 62% of these were responders. 10 actually became seizure-free, and the responder rates varied according to the underlying neuronal antibody involved. Importantly, immunotherapy has now become an established treatment for autoimmune epilepsy and antibody-mediated encephalitis, and it's now part of treatment algorithms. These include intravenous methylprednisolone, intravenous immunoglobulin, plasma exchange. It is filled in what might consider alternative immunotherapies such as rituximab or cyclophosphamide, and in the long run, even mycophenolate and zacaprin. So all of these are immunotherapies. But there's more, and there's more interest in this space. One example is provided by natalizumab. This is an established treatment for multiple sclerosis. It's a humanized monoclonal antibody that selectively binds to the alpha-4 subunit of the alpha-4 beta-1 and alpha-4 beta-7 integrins expressed on the surface of leukocytes. And a case report, which is now 10 years old, provides some interesting insight into the potential efficacy of natalizumab in patients with epilepsy. This was actually the case of a patient with multiple sclerosis, which was well under control, but who had actually drug-resistant epilepsy. And seizures only became well controlled after the introduction of natalizumab. The response was quite prominent in terms of the reduction of tonic-clonic seizures in particular. So this case, as well as the growing interest in the space of neuroinflammation in epilepsy, has then led to the design of a randomized controlled trial, specifically a 6-month randomized double-blind placebo-controlled study to assess the efficacy, safety, and tolerability of natalizumab as adjunctive therapy in the treatment of adults with drug-resistant focal epilepsy. This trial has already enrolled 68 participants and is due to completion on December 16, 2020. So the results are expected very soon. Another treatment for multiple sclerosis, fingolimod, has received attention as a potential treatment in epilepsy. Data from experimental epilepsy models suggest that you may actually have a disease-modifying anti-epileptic effect through different mechanisms. One is a reduction of neuroinflammation. The other one is a reduction of neuronal cell death, a reduction of anti-activation of astrocyte microglia, a reduction of the mTOR pathway activation, increase of myelin repair. The translation of these experimental findings to humans is awaited, and certainly the evidence in terms of mechanism is intriguing, but how these findings translate to patients is yet to be seen. A field where there have been tremendous advances has been the field of epilepsy genetics. We've gone from having a handful of genes in the so-called channelopathy era to an explosion of gene discoveries, which have taken place following the advent of next-generation sequencing. And the explosion of gene discoveries has changed the paradigm used potentially to discover new treatments in epilepsy. The traditional model comprises starting with a compound, be agnostic to causation. You treat the seizures, many compounds are studied, no functional studies are needed, and then you end up with new compounds. Well, the genomic prediction model instead, you start with the disease. There's a precise molecular explanation. You treat the epilepsy, few compounds are studied, functional studies are critical, and there is really a role for repurposed drugs. And the pathway that has emerged through the advances in epilepsy genetics has been the mTOR signaling pathway. These pathways involved in epilepsy associated with tuberous sclerosis complex, but also forms of non-lesional focal epilepsy, as well as epilepsies associated with brain malformations. There are different players in the mTOR pathway. One includes mTOR, which is a target for different mTOR inhibitors, such as with rapamycin and even everolimus. And this is specifically is an immunosuppressant for the prevention of organ transplant rejection, and anti-tumor agent. Is approved for the treatment of subepentymal giant cell astrocytoma in renal angiomyolipoma in tuberous sclerosis complex, and was demonstrated by Jackie French and her colleagues to be effective as adjunctive therapy in the treatment of focal onset seizures associated with tuberous sclerosis complex. And the two arms treated with everolimus interestingly showed an improvement in seizure control over time, as opposed to the placebo arm, where a plateau or quasi-plateau in terms of seizure response was identified, suggesting that everolimus may actually have a disease-modifying effect in patients with focal seizures on the background of tuberous sclerosis complex. Another example of drug repurposing using epilepsy genetics information is the case of memantine. This is an NMDA receptor antagonist. Is approved for the treatment of Alzheimer's disease. And in this case, with an early onset epileptic encephalopathy due to a mutation in the green2A gene, treatment with memantine actually resulted in a significant improvement in seizure control, which could not be achieved with traditional anti-epileptic medications. This is just one case, and the findings need to be explored in well-designed studies, but it's an interesting observation nevertheless, and provides another example of the potential applicability of drug repurposing in epilepsy. Other examples of repurposed drugs for epilepsy include fenfluramine, which has recently been approved by the FDA, anakinra, and potentially quinidine. But these compounds will be discussed in lectures which will be following mine. So where are we heading? There are different approaches to identify repurposed drugs for epilepsy. One is the one proposed by Tony Marston's group, which created a PDE3 database, the prescribable drugs with efficacy in experimental epilepsies. The authors performed a computational literature mining of Medline abstracts containing the name of a prescribable drug, a rodent model of epilepsy, a phrase indicating seizure reduction. This was followed by crowdsourced manual creation of the identified abstracts, and 173 drugs were included. Of this, among the top drugs, there were commonly prescribed agents such as amlodipine, aspirin, atorvastatin, which at doses used in humans were found to have anti-epileptic efficacy in animal models. Importantly, these drugs target mechanisms which are not targeted by conventional anti-epileptic drugs, including transcription factor, membrane receptors, therefore providing hope to improve the outcomes in the treatment of epilepsy, which, as I mentioned earlier, disappointingly have not changed over the years despite the introduction of new anti-epileptic medications. Another way to identify repurposed drugs for epilepsy is the following approach, again used by Tony Marston's group, which is based on a genomics-based drug repurposing paradigm, that every disease can be described by a gene expression signature. And therapies restoring gene expression patterns to their norm are associated with successful outcome. The transcriptomic signature of epilepsy here was based on published RNA sequencing analysis of experimental temporal lobe epilepsy. And drugs reversing such signature were identified using the LINCS database and software analysis. 36 drugs were identified, 11 with published evidence of anti-epileptic effects, such as curcumin, or even anti-epileptogenic effects, such as atorvastatin. Here, another example of precision medicine, ultimately leading to the potential identification of repurposed drugs for epilepsy. It all stems from a mutation in the gene, SCN8A gene, from a patient with an epileptic encephalopathy, which was expressed in cellular models. And the authors, coming from David Goldstein's group at Columbia University, perform a high throughput screening of more than 1,300 compounds using fluorescence-based sodium flux assay, which identified antidrugs that significantly inhibited sodium influx in the mutated cell line. Now, 4 drugs of potential chemical interest, including 3 potentially repurposed drugs, amitriptyline, carvedilol, and nivaldipine, were further investigation and showed concentration-dependent inhibition of the sodium channel. So, in conclusions, despite the introduction of many new anti-epileptic drugs, 1/3 of patients with epilepsy continue to have uncontrolled seizures. There's clearly the need for novel, more efficacious therapies. Drug repurposing is the discovery of new indications for approved drugs and offers the potential of substantial savings in time and cost of drug development. Drug repurposing in epilepsy is already a reality, driven by advances in understanding the underpinnings of epilepsy. Examples I provided include immunotherapies for autoimmune epilepsies or everolimus for epilepsy associated with tuberous sclerosis complex. And there can be future strategies to identify drugs which may improve the outcomes of the treatment of epilepsy, and may facilitate the identification of additional notable agents. This includes literature mining, such as the PDE3 database, and precision-based approach. With this, I thank you for your time. I hope you enjoy the rest of the ALAT 15 conference, and I look forward to your questions. Thank you.
Thank you very much, Emilio, for this brilliant presentation. Clearly, the development of drugs is like swimming in a shark pond. Yeah, questions coming in. So is there any repurposing project for autophagy drugs to treat epilepsy? This is a question more or less related to a compound we're going to discuss later, because everolimus apparently it's known to inhibit mTOR, so increasing autophagy. What's your view on this?
Well, it's in general, mTOR inhibitors do potentially have this mechanism of action. But let me just expand on this, in that I discussed specifically the role of everolimus in one specific condition related to the mTOR pathway, which is tuberous sclerosis complex. What we don't know is whether everolimus may act on other mTORopathies. And so this may include epilepsies associated with DEPDC5, for example, mutation, or NPRL2 and 3, and whether autophagy actually has a mechanism in those type of epilepsies yet unknown. I think this is an interesting space, which I think we should all stay tuned in the next few years. And we have a talk just 2 talks after mine, specifically on everolimus. So I don't want to give too much away on the potential mechanisms of actions.
Yeah, no, fair point. Another question from an anonymous attendee. He has got a special interest in vincpositin, vinca compounds. What's your role in this? Is it a useful treatment?
Well, this is actually an interesting compound, which I did not include in my presentation. Is an alkaloid derived from a plant, it's a periwinkle plant, I think, and I think has a role as a cognition enhancing agent, and it has been explored as an anti-epileptic drug in vitro, showing anti-epileptic properties. It recently was also shown to improve, I think, epileptic activity in a case with Lennox-Gastaut associated with a mutation in GABAB3. And there is also a small trial conducted in Mexico, I think, on suggesting the potential efficacy as an adjunctive therapy for drug-resistant focal epilepsy. Certainly, it's an interesting drug which may act on a variety of different through a variety of different mechanisms, including sodium channels, also GABA channels. So it may well be another example of drug repurposing in epilepsy. And again, I look forward to seeing more data on this particular compound.
Cecily Johansen asked, what do you think would be the most promising approach regarding repurposing when it comes really to mechanism of action? Or if I may add to this, would you expect, particularly from your genetic knowledge, that there will be one mechanism of action? Some people propose that in terms of repurposing, it might well be a cocktail of drugs we have to swallow with multiple agents, statins, inhibitors, COX inhibitors, inflammatory products, all being included in this.
Well, thank you, Cecilia, and thank you, Matthias, for the question. Well, I think what we have been studying so far the so-called low-hanging fruits, where we're looking at specific compounds working on Mendelian epilepsy. So epilepsies due to single gene mutations, but the more common epilepsies display a complex genetic architecture with multiple variants having a small effect size. So to your point, Matthias, you may not be able to just reverse in a way the genomic signature of a condition by using one compound, but you may have to have a more holistic approach, if you wish, targeting multiple mechanisms. And this is why in one of my slides, I wanted to emphasize that, you know, in a few years, we may be ready to adopt a different approach to drug discovery, which takes into account knowing the mechanism of action and then translating that into the discovery of drugs, which may have more than one target.
Yeah, no, away from the dogma of monotherapy. Piero, I think because your talk is really such introductory to this whole session, it would be great if you could stay on till the very end of this session, so that we have a joined up discussion of all repurposed drugs. There's another question which has come up in relation to tuberous sclerosis and everolimus, but I think I leave this for really the next talk when we focus on this. So thank you very much for this introduction to the brilliant introduction to the session.